Hello, my name is Vaidehee Lanke. I'm a bioinformatics student studying at the University of Saskatchewan. My research project is in applying deep learning to predicting plant phenotypes from genomic data. Phenotype prediction using genomic data has important applications, especially when we consider the development of more effective crop breeding programs. Deep learning, a subset of machine learning, can be applied to extract features from genomic data to predict phenotypes. However, the challenge is the scale and quality of paired genotype and phenotype data required to train an accurate model. Work that was previously done in our lab demonstrated transfer learning can significantly increase the predictive power of a model when limited to a small number of samples. And so the research question we sought to investigate was, do the transfer learning and regular prediction task results obtained from Sutherland 2016 lentil dataset uphold to data from an additional location and different years? The genotype data came from 324 samples from the Agile lentil dataset sequenced using exome sequencing, and the phenotypes we looked at came from two locations, Rosthern and Sutherland, and 3 years, 2016, 2017, 2018. We aimed to look at 3 different phenotypes: days to flower, days to swollen, days to mature. For each task, the dataset was split 90% for training and 10% for validation. For the regular prediction task, Model 1 was trained on given genomic data to predict a phenotype, and for transfer learning, the trained weights from Model 1 were transferred to Model 2, and then Model 2 was retrained on the same genomic data to output a different phenotype. The results I will be sharing in this poster come from the days to swollen phenotype and predicting it. So for the regular prediction task, we see for the training that as the number of epochs increases, as the training continues, the loss values decrease quite stably, indicating that the model is getting more accurate at predicting the phenotype. We can see that Rosthern 2016, Sutherland 2017, and Sutherland 2018 follow the trend of Sutherland 2016, therefore upholding the trend we were looking for. And we can see for the validation loss that as the number of epochs increases, it too decreases. However, there are many more fluctuations. When we look at transfer learning, Model 1 that we chose was days to flower, and Model 2 was days to swollen. We can see for the training loss that the loss values decrease stably as training continues, indicating that it is becoming more accurate at predicting the days to swollen phenotype, and that the new data sets are following the trend of Sutherland 2016. For validation, we also see that decrease in loss. However, there are significant fluctuations seen. This could be due to model overfitting, so the model is performing quite well on the training data set. However, it is not generalizing as well to the unseen data in the validation data set. So through this snapshot of our study, we were able to show that the regular prediction task loss trends and transfer learning loss trends from Sutherland 2016 upholded for our data. And when we compared the losses from the regular prediction task to those of the transfer learning, we can say that transfer learning can help us better predict the days to swollen phenotype for all data sets during training and for some of them during validation. In the future, we would like to apply interpretable machine learning techniques to our model so we can understand exactly what features, genomic features, the model is using to predict its phenotypes. We would like to evaluate our model against independent, unseen data to see how our model generalizes. And we'd like to try out all combinations of phenotypes mentioned at the start to see which ones have the most success with transfer learning and to further refine our model. I would like to thank the Plant Phenotyping and Imaging Center at the University of Saskatchewan for its funding of this project and for Dr. Kuzlik for his mentorship. Thank you for listening.